Well, thank you. I'm representing a large team at UCSD that has been working for the last 3 years on the development of what we think will be a transforming product for cancer. This slide describes the clinical need for the product we're developing. The most effective anti-cancer therapies exploit clear-cut metabolic differences between malignant and normal cells. These therapies must be able to kill resting cancer stem cells as well as dividing cancer cells without harming most normal cells. All the targeted kinase inhibitors being developed today and in the clinic act on dividing cells, and when the drugs are stopped, the cancer comes back and resistance inevitably develops. However, the anti-ROR1 monoclonal antibody that we are developing targets a profound difference between these cancers and normal cells, and particularly the non-dividing stem-like cells that survive targeted chemotherapy. The disease we're first working on is chronic lymphocytic leukemia, although we feel the product has much broader application. Experience has shown that starting with liquid tumors that are readily accessible is the fastest way to determine the biological activity, toxicity, and mechanism of action of a drug. However, CLL is itself an important disease characterized by the accumulation of small, mature-appearing monoclonal B cells in the blood, marrow, and lymphoid tissue. It is the most common form of leukemia in the Western world, and is with and as the population ages, there's an epidemic. About 160,000 people in the U.S. and European Union with 16,000 diagnosed per year. As I explained, current treatments for this disease, as almost all cancers, suppress but do not cure it, and therefore more effective and non-toxic treatments are needed. So this slide describes all of the areas that our CIRM grant has supported, right from we did the target discovery, but right through to the final product, which I'll briefly summarize. So what is ROR1? It is an embryonic receptor tyrosine kinase, highly expressed during embryogenesis, but ablated completely after birth. My colleague, Thomas Kipps, discovered that a subset of patients with chronic lymphocytic leukemia, CLL, who were vaccinated with their own CLL cells, developed an autoantibody, an anti-CIR reactive with an antigen, ROR1. And this antibody from the patient reacted with CLL cells, but not normal cells. This was an experiment of nature in humans that had two profound results. First of all, we noted that patients who developed the antibody had a highly indolent clinical course after the vaccination, suggesting activity. But even more important, there was no effect on any normal tissue, indicating that the presence of an antibody to ROR1 is expected to be non-toxic. Further studies on 800 CLL patients shown here showed that 94% of cases express ROR1. And gene expression studies done in collaboration with colleagues in Canada indicated that 6% of patients who did not express ROR1 actually do not have CLL disease. And a simple Western blot with an early antibody shows the profound lack of the protein in all normal tissues as compared to the uniform high-level expression in CLL. However, having made subsequently a monoclonal antibody, we found that ROR1 is a target antigen in many different human cancers, if not all. First of all, ROR1 is expressed by a subset of other leukemias, including acute myelocytic leukemia, B-cell acute lymphocytic leukemia, and T-cell acute lymphocytic leukemia, and by many solid tumors, particularly those cancers that are high-grade or poorly differentiated, such as triple-negative breast cancer, pancreatic cancer, ovarian cancers, lung cancer, the big killers. Shown here is a tissue array where the red refers to the cancerous and the lack of red, the normal tissues. Note that breast cancer, lung cancer, ovarian cancer, colon cancer, pancreatic cancer, skin cancer, testicular cancer, bladder cancer, uterus cancer, prostate cancer, all express it. And remember I told you in the introductory slide, the key to getting cancer cured is finding an absolute metabolic difference or target difference that is expressed by the resting as well as the proliferating cells, and we think that this is a first example. By the way, the tissue cross study that was done here in the laboratory has been completely confirmed by a outside contract research organization approved by FDA. So with this information, there was an anti-ROR1 monoclonal antibody discovery program. At UCSD, originally there were 70 different antibodies produced. Only one had biological activity. This is not immunologic activity, but direct drug-like biological activity. And it sees a unique epitope that's not seen by other antibodies that we are aware of. In a rapidly dividing tumor model, which is often used to screen these antibodies using a transgenic ROR1 on an oncogene, this antibody called D10 caused regression of the splenic enlargement in this leukemia model, whereby the other antibodies, which is indicative of the 69 that didn't work, did not, showing how one epitope is unique. Sorry about the complexity of this slide, but I wanted to show you in another solid tumor model, also rapidly replicating, that the ROR1 antibody blocked breast cancer migration and metastasis in the MB231 line. This figure shown here is an in vitro measurement of invasion and metastasis, also what they call epidermal mesenchymal transition. These are key features of stem cells that also mediate metastasis and eventual recurrence. And without any immunological enhancement, the antibody blocks these in vitro. And in vivo, in a lung cancer metastasis model to the lung, breast cancer metastasis model to the lung, the antibody also inhibits it. But what about the key effect on non-dividing cells? Okay, so in order to analyze this, we developed a parking model in which human primary chronic lymphocytic leukemia cells are put into the peritoneal cavity of profoundly immunodeficient mice. And they actually don't proliferate there, but they survive in a resting state. The same animals were repleted with human bone marrow stem cells and form human B cells and T cells at the same time. So each animal is its own control because it has the leukemic B cell, the normal B cell, and the normal T cell. Then the antibody, now fully humanized, which we call 961, was administered. So the antibody depleted 98.5% of the non-dividing primary human CLL cells, but had no significant effect on human B or T cells, confirming the tissue cross and also providing us with a sense that our drug will be safe. So this describes all of the process of development of a monoclonal antibody, now a very, very mature technology when the proper antigen and the proper epitope is developed. And every one of these studies was paid for by CIRM and would not have been done otherwise because of the riskiness of the project. So the monoclonal antibody creation, the humanization, the producer cell line, the pilot manufacturing, the farm tox, and the pre-IND meeting. And we are now in our last 6 months of the grant, working on the things noted in red for an IND submission early next year. And the primary target, again, will be CLL because it's very, very easy for us to measure not only survival of the cells, but the RNA expression levels, cell surface expression levels, and seeing the toxicity. And I think this will be profoundly interesting. So in summary, this antibody now targets cancer cells, but not normal cells. And to the best of my knowledge, it is the only absolutely cancer-specific monoclonal antibody. CLL is the prototype, but also found on most liquid and solid tumors. It acts like a drug. It is cell cycle independent, inhibits both growing and quiescent cancer cells. At least in mice, it controls cancer cell invasion and metastasis. And as I said in the first slide, we believe that the human experiment of nature that prompted the project points to safety and efficacy. Clinical trials in CLL will begin in spring 2014, phase 1, and if safety is noted and hints of efficacy, we hope to progress these to solid tumors in the coming years. And obviously, there's a huge therapeutic market for drugs of this type. So I'd like to acknowledge many people in the team at UCSD that working on this. I've been the leader because I've been looking for my whole life on targets in cancer cells that don't hit normal cells, but also Catriona Jamieson, who's a co-principal investigator, Thomas Kipps, who's the ROR1 project leader who discovered this, and Ida Deichaite in our business development group. And again, if we didn't have the CIRM grant, this would not have happened, so I'm very grateful for that. Thank you. No, it's not. It can kill cells via complement mediation, but it actually, ROR1 is a receptor for Wnt5a, which activates the alternative Wnt pathway, which is very context dependent in lymphocytes, involves NF-kappa B and a whole host of other factors. The antibody is rapidly internalized along with the receptor, and therefore the receptor for the essential survival and growth factor is no longer there.